So our final speaker of the session before moving to the round tables is Jerry Cox from Sanofi Genzyme. He's a MD PhD, and many of you will have met Jerry before, and he was conducting some very interesting clinical analyses at last year's meeting, and many of you will have participated. And he's now going to update you with the sort of outcomes from those studies. The idea being, how can we decide on what should be measured clinically in a clinical trial, what will be useful things to measure in the patient population, so that we can try and maximize the chances of success for clinical trials in the future. So I'm going to hand over now to Jerry, who can update us.
Thanks, Fran. It's a pleasure to be here to speak to you. I've been waiting all year. We had quite a good session last year evaluating patients for the term we're using is clinical trial readiness. A lot of people think that when a drug becomes ready for use to be studied in patients, that you just go ahead and give it to the patients, but you actually have to know, what are we going to be looking at to know whether the drug is actually working? And so last year we carried out a series of assessments over, I think it was a 2-hour time period, try to get a sense of whether there were some types of assessments that would be of the type that would be done in a clinical trial that could show whether or not a drug worked or not. Are you advancing slides or? Okay. So last year we brought together a group of 12 patients, 10 with late onset Tay-Sachs and 2 at Sanofi. And the purpose of this slide is just to highlight that both of those conditions are caused by the same enzyme deficiency, just different subunits of hexosaminidase. And in each case, there's a particular lipid called GM2 that accumulates in Sanofi and Tay-Sachs. It's primarily in the brain, and that accumulation causes the nerve cells in the brain to not function properly and lead to clinical symptoms. So oftentimes we do consider them related conditions, and they have a lot of overlap. So that's why we're looking at both. And before you actually start to do trials for patients, you want to do a little background about the disease. So what do we know about what causes Tay-Sachs and Sanofi, what's responsible for early onset or late onset? And a lot of the early studies pointed towards the amount of hexosaminidase activity as being really critical in determining the severity of the disease. So in infants who have very little or no activity, the disease presents at a very young age, and the amount of GM2 accumulates very quickly. In other individuals that have a little bit more enzyme activity, the GM2 accumulates more slowly, and that's why those symptoms don't occur until childhood. And then there are even other patients who have more residual enzyme activity where symptoms don't begin until adolescence or sometimes in adulthood. And so we believe a lot of the underlying disease is related to the amount of substrate and the speed that it builds up. Now, about 10 years ago, Ed Kolodny and some neurologists got together, and they studied a group of late onset Tay-Sachs patients, and they were characterizing what we call the natural history of the condition. So in the average patient, when do different symptoms occur? And what they found was that in adolescence is when a lot of the patients began to complain of issues with balance, some muscle weakness, difficulty climbing stairs, even in some cases psychosis or mood disorders. And then over the next decade or 2 decades, this would progress to the point where now other organ systems became affected, so eye movements, speech being more difficult to understand, what we call dysarthria, and continued progression of the really lack of coordination of motor control, what we call ataxia. And that includes unsteady gait, poor balance, difficulty with precise movements using your hands. And then over time, it progresses more and more until late adulthood. So we look at this and we say, okay, there's a lot of medical issues here, but if you were to have a new drug, what would you actually look at? How would you quantitate this? And is this really reality? Is this what other patients are complaining of at those ages as well? Because this was a small sample size. And so we thought last year it would be really good to almost start with a blank sheet of paper and conduct some focus groups with patients and caregivers and just ask open-ended questions about their condition and what their complaints were, what mattered most to them. And with those discussion groups and reviewing the literature, we also picked some assessments. We made educated guesses based on these types of symptoms, what types of assessments might be sensitive to show degree of severity and how frequent the changes are. So last year we did what's called a cross-sectional study, meaning we have a group of patients that we look at one point in time, and we're going to look across that whole group and try to reach some conclusions about the disease state. And what we were specifically doing is wondering, were there measures that we could look at that could actually grade the patients from mild to severe? Were there other functional assessments that would give us an indication if some patients were having more difficulty doing a particular task versus another? And the reason I mention that is to get a drug approved by the FDA, there's a very simple criteria. You have to show that there's an improvement in the way a patient feels, functions, or survives. So survival usually is not an option for a lot of clinical trials unless you're dealing with a rapidly progressive disease like cancer. Symptoms, people can vary in their symptoms over the course of a day from one day to the next. You really need a large number of patients in order to show an effect on symptoms. And then there's function. And so there's a lot of tests that I think we can exploit that look at different aspects of functioning, walking ability, speaking ability, using your hands, thinking. And that's what we focused on last year. During the roundtable discussions, I think Ala Hamed is here, who conducted some of these patient interviews, and will give some feedback on what we learned there. But the issues that the patients really seemed to highlight were difficulty walking with an awkward gait, fear of falling, because it's very difficult to catch themselves with a poor balance, and difficulties with speech and communication. Next slide. So we did this study. We put together a protocol where we specify what we're going to do. We send it to an independent group that reviews it to make sure it's ethical. That's called an IRB. We have an informed consent that patients read. And then if they agree to all the conditions and terms, they sign it. And then we go ahead and we do the study. And I've just listed here several of the items that we looked at. We evaluated some severity scales, either just simple scales or scales more related to ataxia. We had an assessment of mobility, and I use that as a general term. It's not just walking, but actually getting up out of a chair, transferring. That's also a form of mobility. Hand dexterity, how well can someone use their hands to do a fine motor task. We looked at tremor. Speech clarity by reading a standardized passage called the Rainbow Passage. Executive function, a type of thinking. This test is called the trill-making test. I call it connect the dots, and you'll see why in a minute. But it involves some thinking. They're not just any dots. You have to connect them in a certain order that requires some thinking. And then we looked at some quality of life measures as well. So the first thing we do when we do a study is we ask, who are we dealing with? What does the patient population look like? And you could see that the average age was mid-adulthood, 47. Just over half the patients were males, all were Caucasian, 40% were Ashkenazi Jewish. Of the 12 patients, 10 had late onset Tay-Sachs, 2 had late onset Sandhoff. A highly educated group compared to a lot of other studies with college education. You can see majority of patients had been married, either currently or past, had children, and just over half the patients were not able to work. Then we look at the disease, and what I find really striking here is that from the time that patients told us they had their first symptom to the time they got diagnosed, on average it was about 18 years. and that's just unacceptable. I mean, the disease is progressing during that time period. So we see that with a lot of other lysosomal storage disorders that we treat as well. And then from the time of diagnosis to the time we saw them last year was another 12 years. So we're dealing with a population that's had symptoms for 30 years on average. And so that's if you will, a skewed group, because that group is going to look quite different than perhaps newly diagnosed patients or patients that come to medical attention because of an affected sibling. Previous slide. We also found that just under half the patients required caregivers. Nearly all the patients used some kind of assisted device for ambulation, either a wheelchair, a walker, or knee brace for stabilization. And about half the patients had dysarthria, or difficult to understand speech. So the first thing we did was we wanted to look at the disease severity. And there's a very simple scale that we use that just goes from completely normal to as severe as you can imagine on the left, with just some intermediate steps going from mild to moderate to severe. That's a pretty crude scale, but I think there is an element of validity to it. On the right is a more specific scale related to ataxia. And this looks at different areas that ataxia can manifest. So gait is the walking quality of walk. The knee tibia test is the lower motor coordination. Finger to nose is upper motor coordination. Dysarthria is the speech, and oculomotor is the eye changes. Next slide. One of the first things we want to know is whether older patients had more disease severity than younger patients, and we found that that wasn't the case. We had some older patients that were mild, some that were more severe, and the same with the younger patients. So neither of those scales seem to correlate with age. Next slide. But reassuringly, the physician global impression correlated very well with the ataxia. So what that says is that what the physician is actually seeing is the ataxia, which we're measuring specifically, and the two of them are kind of linking up. So what we think is the disease severity is showing up as the ataxia. Next slide. And then we wanted to know, well, this ataxia scale, does it correlate with any of the functional measures that we did? So this get up and go test, patients rise out of a chair, they walk 10 feet, they come back and they sit down. And normally you should be able to do that in about 12 seconds or less. But we found that on average, patients took about 20 seconds. Many of them were using walkers. And we also found that 5 patients couldn't do the test because they were either in wheelchairs or they couldn't get up. So that was important to know, because when you have an assessment for a clinical trial, you'd like to have all the patients be able to do the assessment, and here about almost half of them couldn't do it. Next slide. We also looked at the fine motor coordination, putting pegs into a peg board and taking them out. We found that that was also, on average, at the lower end of the normal range, and it also correlated nicely with the bar score. About 40% of the patients registered as below the normal range for doing that task. That's a time task. We also had patients trace spirals, which looks at tremor, and that also correlated with the ataxia score. Next slide. And we looked at speech, just in the interest of time, that also correlated with that simple bar score we looked at. And then the trail making test, we have people trace, connecting the dots, either sequential numbers or alternating numbers and letters. And that's also a time test that involves thinking. Next slide. Back one. And those tests also correlate with the bar score. So a lot of the functional tests were correlating with the disease severity score that we used. And I think what I would just say, what we learned last year is I think there are going to be functional tests that will be very informative for clinical trials. Some of them, I think, now knowing the patient population better, we may need to think of doing some different tests, like getting up out of a chair is very difficult. So this year we did a simple walk test. We added that. Last year, we just had a very simple scale for looking at speech. It was like a 5-point scale. This year, we actually had patients recite single words as well as sentences as part of a more standardized speech assessment. And I just want to thank everyone that participated in the assessments last year and this year in particular, to NTSAD, Sue, for helping to corral everyone together for these sessions and putting together the logistics, and also the patients who participated. We had very good turnout this year again. You heard that we were doing the sessions this morning. We'll be doing more tomorrow. We have even new patients coming in this year. So one of the things we'll be looking at this year is whether any of the scores that we measured last year are now different this year, and if they track with any kind of change in disease status. So thank you very much.
Okay, well, thank you very much indeed. Again, I think we've gone from everything from very basic science of how to put things into the mouse nostril through to clinical assessment